Transcription is the process of synthesizing an mRNA molecule from DNA to be used for protein synthesis. In this process, an enzyme, which is called RNA polymerase, the enzyme RNA polymerase copies the instructions for the protein from the DNA strand, and that same enzyme uses those instructions to make the RNA. This is the transcription process. The DNA strand that carries the instructions for the protein synthesis is often called the informational strand because it has the information. So I'm going to just write a little informational strand right here. GCACATT from 3' to 5'. And so we're going to say that let's just imagine that this piece of the DNA right here contains information about synthesizing some sort of protein. So this is where we have the protein synthesis information. And this is the part of the DNA that we need to copy in order to take the instructions to synthesize this little protein, take these instructions into a ribosome. This, again, as I said, is often referred to as the informational strand. Now, the RNA polymerase doesn't copy this informational strand directly. It actually copies the complement to this informational strand because we want to create a replica of this informational strand. We don't want to copy this strand. We want to copy the copy of that strand. So what gets read is the complementary strand to the informational strand. The complementary strand referring to the complementary base pairs here. This strand, this is actually the one that is read. This is called the template strand, because this is the strand that is being used to synthesize the mRNA. The mRNA is then synthesized based on that template. And in doing this, we will create a pretty exact copy of the informational strand where the DNA instruction or the protein instructions exist. So this mRNA strand that we're going to synthesize is GCACAU, remembering that mRNA has U in place of T. So this right here would be the mRNA strand. And then this is an mRNA version of the informational DNA strand that contains the information for protein synthesis. So this portion, this mRNA strand, would leave the cell nucleus and then would travel to a ribosome where it would work together with the tRNA molecules to assemble the amino acids in order for protein synthesis.